This is CheloniaCast, episode 78. Today we are joined by Jordan Donini, who's a professor of biology in the School of Pure and Applied Sciences at Florida Southwestern State College. Jordan wears many hats in the turtle world. On the one hand, he's an incredible researcher, focused on a lot of different aspects. I mean, the general ecology, reproductive biology, ecophysiology, and conservation of turtles. He spans a lot of different fields. He's published in journals like Chelonian Conservation and Biology, Amphibian Reptile Conservation, and a number of others. On the other hand, he's a great teacher, a mentor, and a really creative thinker. I think that comes through in a lot of his publications. He's finding a lot of really interesting things to look at that we're going to dive into today. He's inspired many students through research with his lab, the Southwest Florida Turtle Project, and the through the Honor Scholars Program at FSW. So we're really excited to talk with him today. I think he's inspired all of us as the hosts. So this is going to be a really fun conversation. Thanks for joining us.
Happy to be here. Always happy to talk turtles with fellow turtle nerds.
Sweet. Someone want to, Jack, kick us
I
off?
guess on the topic of turtles, what sparked your interest in turtles? When did that begin?
Oh, really, really early. I was a dinosaur kid, like I think a lot of us can probably relate to. Um, but non-avian dinosaurs aren't really around anymore, so it was kind of hard to study those or to observe those. So I went to the next best thing, which was extant reptiles, and turtles just always were fascinating to me. I was a Teenage Mutant Ninja Turtles kid growing up, so I had that love going for me. And then my first ever pet reptile was a red-eared slider, so that led to just the interest continuing to be nurtured. And once I finally got to the age where I could start studying and doing research on things, turtles really just took the lead, just because there was a lot of questions to be asked about turtles that hadn't been answered, and I was hoping to kind of fill in that niche and help continue to inform people on all of the cool things that we didn't know about turtles yet.
That's awesome. I'm curious. This is one question I've had is just, I mean, who were some of your early influences in turtle conservation research? I mean, you're really well connected with people, so I'm just curious about that.
Yeah, so I'm a little bit more non-traditional, in that, like, when I was getting out of high school, like, I didn't even know what, like, the word ecology meant. Like, it was a slow build before I really got into a lot of the nitty-gritty of it. So, I was like, a lot of people were like, Steve Irwin was kind of the trigger point, right? He made it cool to go out and catch herps and to observe herps in the wild. Um, Down here in Southwest Florida, we did have a local herpetological society, the Cloaca Herp Society, which was a mixture of like captive keepers and researchers and field-based folks. And one of my good friends, who I was in high school. Had just started working down here in the area. Chris Lekowitz, I think some of y'all have met him before. He's kind of one of the Graptomes experts out there. Um, yeah, he was one of the first like real like herpetological researchers I was able to connect with. And with him, I was able to see, oh, this is the kind of stuff you could do as a job. This is really, really, really cool to see. So that was one of the first like actual scientists that inspired me. Once I actually started getting into the literature and things like that, right? Archie Carr became a huge influence. I mean, I feel like anybody that's ever looked at turtles, Archie Carr is up there. Peter Pritchard. I was fortunate enough to meet Peter Pritchard at the All Florida Herpetology Conference as an undergrad, which was awesome. He did like a sort of open symposium type of deal, talking about the recent changes to collection policies of turtles in the state of Florida, along with some captive turtle folks. Jerry Johnson was on the panel as well. But it was really cool to actually meet Peter in person and hear some of his wisdom. So I think, as many of you can attest to, he's been an inspiration to all of us. And then my first ever real undergrad advisor in terms of herpetology is Dr. Phil Allman, who's mostly a sea turtle fella, but has done a lot of work with gopher tortoises and diamondback terrapins, as well as some other species. He's the one that kind of got me start on my actual data collection career. We did some work with emided turtles and urban ponds at the Florida Gulf Coast University campus where I went to undergrad, and that was my first real research project that then spanned on to where I'm at right now.
That's awesome. So that mention of Dr. Allman or Phil Allman, that there was one of your publications was pretty interesting. I was reading through that, but the on the gopher tortoises, and it seems like their reproduction is actually occurring in the winter and for a longer period. I mean, what is that about? Why is that?
Yeah, there's still more to be found out on that. Um, but like, there's some people that kind of disagree with what we came up with because we were only able to document the hormones. We didn't actually document the clutching. But Ray Ashton had palpated gopher tortoises with eggs in South Florida in November, so it's been documented before. It seems like they almost have a bimodal reproductive cycle, where they kind of go off during the standard sort of spring, summertime. But then in South Florida, it seems like they also kind of hit during that November, December period as well. What causes that? Still to be determined. But as those of you that came down and visited for our Boxing Day survey know, I mean, Southwest Florida in December is beautiful. It is like we had some cold weather this year, but it was for like a week maybe. Like we're still getting 70 degrees in the daytime, which is plenty of enough heat for tortoises to be out moving around. I think y'all even got to see some tortoises moving around when you came down and visited too, uh, on in the middle of December. So year-long activity, basically. The acquisition of energy probably plays a role into that. Could it be an adaptation with sort of shifting towards more tropical climates, or could it be just phenotypic plasticity? All on the board. People far smarter than me will have to figure that question out, but there's a lot of reasons why they could be continuing that or expanding that reproductive cycle down here. But there's a lot of hypotheses.
I was thinking chicken turtles when I was reading through that. It's similar to the Florida, I guess. But then
100%.
yeah, I thought that the chicken turtles, it was universal that they are producing eggs later in the year, November, December. Apparently, that is mostly just in southern latitudes.
Yeah.
So
Yeah.
that's
Yeah, like the Western chicken turtles are like more akin to like a standard reproductive cycle. If I remember some of McKnight and Legon's papers, it's sort of like a single sort of clutching period. And then Florida chicken turtles, it's pretty bimodal. Like, but I think it may even be more than bimodal down here, because we see them sort of start like in October. They kind of go October through November, December, kind of stops, and then January, February, into the early March stops. And I need to find records. I've been talking to like Kim Titterington and other folks about looking at records, but I'm sure I've heard of them spiking again March, April. So I think there could be more of a sort of uh, continuous reproductive cycle with some abrupt stops throughout. But that's my next hopeful big project. Is I don't think y'all got down for the first day, but we have a chicken turtle site. We just have to figure out the best way to get hands on them without getting destroyed by giant alligators that are in the same habitat with them. But that's my hopeful next part, is to throw some radio transmitters on some chicken turtles, take ultrasound and hormonal data through a year, just to see when those peaks and valleys are in their hormonal concentrations and when those eggs are actually developing. That could be a little tricky though, because some of you may know, like, they retain eggs for like really long periods. So are we looking at a new clutch, or is this still the same clutch we saw last month? So there's some issues we'll have to get through, but I think for Florida, there's basically nothing been done on that, so we need to really get out there and investigate it.
That's awesome. Yeah, the chicken turtles, something about that. They seem to be we Jack and I had a run-in with I guess we were with Parker Gibbons a while back, and he was going after a chicken turtle and ended up uh, brushing a gator. So I guess there's a relationship between that.
Swam,
See,
it swam between its legs. Like it was like a huge gator too. That's And all we found that day was sliders, so it wasn't like.
Yeah, we've got some ideas on how to do it, but I work with predominantly like fresh green undergrads. A lot of them have never been in the field in their life, so there's sort of a balance of safety and conducting the research we have to consider, because if these folks have never been in the water with an alligator, we don't want to put anybody at risk of a situation where they would not know what to do. We haven't had too many crazy encounters with alligators in our studies, but we did get about a 7.5 footer in a fike net last year, so that was fun.
Yeah. How did you get it out of the fike net?
Well, it basically destroyed the fike net, so we just cut it open and got the animal out. It twisted itself up so much. It was about, like, gator itself was probably about 80 to 100 pounds, so it was a pretty good sized gator. But then there was like an additional 100 pounds of turtles and fish in the same trap. So getting that trap out of the water was crazy. It's like I got into the water and saw the trap was sunk, and I'd assumed like there was just a whole ton of turtles in it because we'd never caught gators at this spot. And then I pulled the trap up, and this gator's head just pops up and looks at me in the trap. I'm just like, oh, jeez. Okay. Okay, let's reassess the situation here. We were able to get it up, and gator got back in the water, no problem. But yeah, other than that, it was we just basically had to destroy the trap to get it out because the trap was no longer functional. It completely destroyed the inner funnels. So
Dang, the fun part of being in the field, you never
Yeah.
know what you're gonna get, right?
yeah,
Well, I'm curious. I mean, you mentioned the reproductive cycles of turtles are really interesting, and I think kind of it seems like I'm underappreciated almost. And you've
yeah,
done a lot of work with multiple species on
mhm,
this. It's like you can use this set of hormones to get an idea for the patterns and how the turtles are going about reproducing and what goes into producing those eggs and the
mhm.
sperm and all of that. I'm just curious if you could talk to how using hormones, why is that important in conservation biology in general?
Yeah, well, it's got a number of uses. I mean, my primary interest and application is just looking at phenology and timing, because you can look at peaks and valleys of hormones and basically predict when another clutch is going to come or when another clutch is likely happening, at least at the population level. Even at an individual level, you can look at things like progesterone, which is highly associated with ovulation events, to kind of get an idea of when a clutch is going to be coming. But you can really kind of timestamp when peak levels of reproduction are occurring in populations if you look at the hormones. Testosterone is a big one. Estrogen, in the form of estradiol, are big ones, and those two are often tightly connected, because if y'all I know how much you know about the background of like endocrinology. Testosterone gets aromatized into estrogens, and that's one of the main drivers, like in TSD, right, is the activation of that aromatase that turns the testosterone to estrogen, at least the feminization. So when you see kind of like these dueling peaks of both hormones, or maybe testosterone's going lower and estrogen's going higher, that can give you an idea. Okay, they're starting to aromatize these things, and that estrogen, its main job in the reproductive cycle is actually triggering the deposition of egg yolk in the form of vitellogenin, which is the major protein that is found in the yolk that gets deposited in the egg. So when you see estrogen go up, usually that means vitellogenin is going to start to go up, and that vitellogenin is then going to get transported into the developing eggs.
Right, and so that those metrics, I mean, those variables, you can use that to get an idea for the cycle and
Yeah,
how that is playing out on
100%.
a year-to-year basis.
Mhm.
And turtles are fascinating because it seems like there's a lot of different variations in terms of how they accomplish like vitellogenesis
Yeah,
and when they're producing eggs, if it's
yeah,
year round or if it's
yeah,
seasonal. I guess with respect to terrapins, because you've looked at this a lot,
yeah.
how do the reproductive cycles of terrapins compare to maybe North American turtles broadly, and what is interesting about them in that respect?
Yeah, so if you paint them with a broad brush, right, there's kind of two general reproductive cycles, at least talking about female cycles. You've got prenuptial and postnuptial cycles. So basically, is this stuff happening after the breeding season, or is it occurring prior to the breeding season? You can kind of generally split it into those two categories. There's obviously intermediates dispersed about, but most of your temperate cycles correspond more with those postnuptial cycles, where you sort of have this sort of senescence period, or excuse me, not senescence, but quiescent period through a lot of like the late summer, but then fall kind of picks up and you start to see the redevelopment of eggs and things of that nature, and then proceeds into the full reproductive section of it. Terrapins in general tend to follow that temperate cycle or postnuptial cycle as a whole, with some modifications depending on where they're at. So when you think about terrapins, like in the north, up in New Jersey, Maryland, etc., right? Typically, there's going to be maybe some reproduction in the late fall, but then they usually go inactive in the winters, right? Just too cold. They're not really going to be out and about. Then spring hits, breeding kind of goes off crazy in like that Marchish area, and then April. Up north, they really don't start reproducing for the most part. Again, there's variability in that till like sort of like that maybe late June, but usually like July, August, as we're going to see peak reproduction of those, at least in terms of nesting, where they're going to actually start coming onto land and laying their eggs. Then they lay their eggs, sometimes one, sometimes two. I don't know if they've documented triple clutching up in the north, but then after that last clutch, they kind of go quiescent, and then probably late summer, early fall, the follicles start to redevelop up again. They usually hold them steady during the winter, and then again, spring picks up again, and you see the follicles start to develop, and that whole cycle starts over again. That's kind of the major idea of that postnuptial cycle. And terrapins seem to hold mostly true to that, even as you go down. Louisiana terrapins seem to do about the same thing, which I studied them. New Jersey terrapins, I assisted on a study with that cycle and those animals. And then Florida terrapins generally held the same thing, but the concentrations of their hormones and the egg yolk protein actually remained higher elevated in the sort of off seasons compared to some of those more northern populations, which again could be corresponding to the lack of an inactivity period. They don't really go inactive down here. Like, if I would have had time, I could have thrown traps down here when you guys are down, and we probably could have caught 4 or 5 terrapins on a night if I would have had time to set traps, because they're just active all the time. You can always find them if you know where to look. So that increase in activity may mean an increase in energy, and they can then allocate that energy to development of follicles, which could basically mean they might be able to reproduce all the time. But it's more than just having the energy. There's got to be other triggers that trigger when reproduction and follicle development occur. So I think it's likely that terrapins probably disperse from the northern latitude as opposed to from a tropical upwards, and they're sort of retaining that or conserving that postnuptial cycle as they go down. But now, as things are sort of changing around them, we may be seeing them change their cycles to kind of fit where they're at, with tropical occurrences allowing them to possibly do it more often or extend it in. And we do think that there is extended reproduction, although my papers had not been able to fully document it. Like, we find hatchling terrapins in February, um, and in South Florida we know they're not brumating, right? We're not overwintering in nests because it just didn't get that cold. So is that based on the fact that there's eggs being laid like in like late fall potentially and leading to that 2 or 3 month incubation period? We don't know yet. Or is there some sort of other idea with hatchlings where they're estivating down here that we just don't know enough about yet? But I think there's definitely a degree of extended reproduction, because they start basically late April down here, if not sooner, and they go through August with 2 to 3 clutches at least. So they are definitely up in the ante when it comes to clutch production and over time down here.
You touched on I guess a little bit on the evolutionary like idea of the species, and where did they originally come from? Like, if you were saying that the retention of some traits from having evolved up in the north and moved south, I was under the impression that they originated more from like the Gulf, like of Mississippi area, and then spread eastward.
Sure.
So
I guess when I say north, I just meant above Florida, not
okay, that's
necessarily
what I
up there.
wanted to. I was like, wait
Yeah.
a second. Were you?
When I talk, I discuss everything in
yeah, I was
Florida.
like, okay, your parameters are Florida because you know
Yeah.
everything is north to where you.
Exactly,
But
especially where I'm at.
I'm thinking like New England, you know, northeast, you know, northern diamondback terrapins, and I was under the impression, I was like, wait a second, that but the trajectory of the species was from the Gulf region around
Yeah.
Florida up north. So
Yeah.
yeah, it's a bit tangential. I just
No, it's good. And as you, I'm sure, are well aware, it's likely that they came from a common ancestor with graptomes very recently, and that basically spread and exploited those coastal regions, and then was able to spread from there.
Yeah, actually, just asking about that. So, I think if I'm correct, it's graptomes and malaclemes, you know, separated. I think it's some 3 million years ago, if I'm correct. Yeah. So, is there any like comparable things about their seasonal activity? I know it's different because it's a coastal species versus you know a riverine or lake dwelling species, but
yeah. Well, in general, the reproductive cycle of terrapins seems to follow that typical temperate cycle of most of your amidid turtles. At least your typical like basking species like graptomes, very similar. At least most graptomes. Again, there's many graptome species we haven't described the reproductive cycles are, but Flavimaculata follows a very similar pattern um, to terrapins. Even hormonally, it's not too different from the northern species, at least. I think Graham 2015 looked at the reproductive cycles of Flavimaculata. I think it was Flavimaculata, and showed similar peaks of sex steroids that we would see like in our northern terrapins and our Louisiana terrapins. But obviously the Florida ones are quite different. But it seems pretty similar and conserved, very comparable to that of like chrysomes. What's weird, what's weird to me is there's not been a whole lot of like studies on wild populations of trichomes in terms of their reproductive hormones. Like I'm like that's like the study turtle, and nobody's really like just looked at wild populations reproductive hormones. But I've obviously looked at like egg production and output, right? But the hormones haven't been studied. I'm like, man, we derived our assay from trichomes vitellogenin, but nobody's actually looked at wild trichomes vitellogenin cycles and things like that. So it's just really interesting.
That's a low priority species for what people want to work with.
Yeah.
Everybody's like, yeah, no, I don't want to work with sliders. I want to work with like literally anything else.
But you would think before you went out like an idiot like me and started doing it with hard to find species, someone like me would have done it with like a common species I could have caught in a roadside ditch. But
yeah.
When I was running the population viability analyses for the pond turtles and sliders at my local creek, I was trying to get the like fecundity, annual fecundity for the sliders. I realized there's not. I mean, I had to pull from a lot of different data sets, and I came up with like 25, 27 eggs per year per individual. But I'm curious. I mean, I'm not convinced about that number. I don't know if you've come across a fecundity metric for them. That's kind of a random question too.
Yeah, that's a little out of my wheelhouse in terms of
Yeah.
modeling stuff. But yeah, I haven't personally. I mean, I've read some stuff on some of the neotropical sliders where they're popping out like maybe 4 or 5 clutches a year, which is insane to even think about. So who knows what the fecundity metric is there? But yeah, I haven't looked at anything specific.
That's it. Yeah, that's a random kind of thing. Well, I mean, so okay, the cycle for the postnuptial cycle is like you've kind of characterized that. What I am curious though, because so the terrapins in the South in Florida they have kind of an extended reproductive period, but they're not. It seems like they're not completely perennial.
Right.
Are there turtles? I mean, what does the cycle look like in a turtle that is actually fully perennial reproducing? And
well, I mean, so there's kind of 2 ways to describe that, right? You have like continuous reproduction, And then there's acyclical, right, where it's like, haha, we can do it all the time. And to my knowledge, there's nothing that's ever been documented as truly acyclical, because it takes a lot of energy to produce eggs, right?
Yeah.
But continuous reproduction is like, okay, in a population, you can find one of these individuals nesting any point during the year, but it's not like you're going to find the same individual every month laying a new clutch. It might be every 2 months, possibly, or every 3 months, you might find the same individual multi-clutching, but it's not really like continually every month, boom, 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 like some of your invertebrates or other organisms may do this sort of quick, fast throughout their lifespan as much as they can. We just don't see that, or at least we haven't documented it yet. Now, I know in some captive populations of turtles that have constant acquisition of energy and probably over energy, like you can almost see acyclic reproduction. I know somebody who kept wood turtles, and they were like, "Yeah, I've found my wood turtles, at least his group of wood turtles, nesting in every month of the year, and being kept in Florida." And it's like, well, that's interesting. Wow, every month of the year you could find a clutch of eggs from wood turtles. Very interesting. So, yeah, again, it's there's probably some relation to the energy acquisition, like being able to eat all year round and get good energy all year round, that allows them to do this. But yeah, nothing yet on a truly acyclic cycle.
Are the Florida box turtles close to that? Because I mean, you found eggs in the most months, right?
Yeah,
Is that
yeah, yep. Earliest ever on record, and yeah, the I think we've documented the most, at least minimum amount of clutches in Florida box turtles. Yeah, we have January starts reproductive nesting season in Florida box turtles. So they started a month ago. So we see them in January regularly. We can start feeling very big follicles in December. So I think it's just a matter of time before I find one gravid in late December and add to that record. But January is the earliest on record right now for, I think, for any of the terrapini species. Although, I don't know about some of those true tropic species down in Mexico and whatnot when their seasons start. But at least in the United States, definitely the earliest is January. And you can find gravid box turtles from January through August, with kind of a cessation in September through December. And so it's basically eight out of 12 months you can find reproducing box turtles down here in terms of eggs.
wow?
Yeah.
So it's. I mean, it seems like that's almost at that acyclic.
Very close.
Yeah,
It's like
yeah.
4-month break and then boom, and multiple clutches in that time frame. We at least 3 with possibly 4 clutches in some of them.
I'm just curious. You're talking about like obviously the seasons for gravid turtles. So you're saying there's that cutoff at around August, and then there's another 4-month period. I wouldn't anticipate the conditions, like the weather would be that cold yet. Why do you think that is?
That's a great question. Again, another one that I don't have an exact answer to, except for maybe there is some of that conservation of postnuptial cycles where they need like an inactive period, or there's some sort of environmental trigger or cue that needs to help trigger vitellogenesis. Right? A lot of these animals evolved from organisms that underwent hibernation, or excuse me, not hibernation, but brumation periods. And brumation is often associated with the triggering of some of these hormone cascades. Sometimes they need sort of a cool down period. As if you talk to captive keepers, right? Keep Russian tortoises, you got to cool them down to really get egg production most of the time. Not always the case. So there's likely just some sort of conservation of that quiescent period that maybe they need, maybe they don't, but it's still actively part of their cycles right now for whatever reason, because it really doesn't get that cold. Like we were down here surveying for box turtles in December, and we didn't catch that many, but we still caught a lot, and they were not exactly inactive per se. Like some of them were out moving, some of them were buried under some leaf litter, but in general they're still active and moving. We still find them with full guts in December all the time. So I don't have an answer for that, but it's definitely a really intriguing question to think about.
Is there an advantage of having an extended reproductive period versus like a true perennial one?
It's a good question. I would think, I mean, obvious advantage, right, is having at least extension means you might be able to span multiple different temperature regimes, right? So especially with things like climate change, right? Maybe earlier or later, the incubation temperatures may be variable enough to help with the sex ratios. I have a friend who's actually doing some research on that right now, Dr. Sean Duty. He's basically looking at a lot of like those continuous nesters, like peninsula cooters, and how you can literally find a peninsula cooter nesting any month of the year in Florida, and looking to see if like location of the nest varies between months in terms of like exposure to solar radiation, and you can use that to kind of calibrate potential temperatures of the nest. So it could be something along those lines, but I don't know about an advantage to having like a fully, a cyclical sort of situation, if there'd be any more or less advantage to that.
Also, I mean, this is kind of nuanced, but I wonder how much energy does a turtle put into finding the right nest site?
Right.
Is there a trade-off between it's like yeah, obviously there's energy going into the eggs and a lot of energy at that, but also I imagine that there's a lot of movement and that sort of thing that goes into finding the right site and just brain power, if you will. So is that something? I don't know how
Yeah.
you would test that necessarily.
Now, what is it, like a third of glucose in a system goes to brain function or something like that? So it's like a huge amount of energy just to basically get your body moving to find those nesting places. And some of these animals are moving far distances from their aquatic habitat to nest, right? Like I'm sure like your river turtles that some of y'all work with, they're probably just going up onto banks. But some of these like pond turtles, there may not be good habitat for several hundred meters away from the pond. And if you're doing that middle of the summer, where the threat of desiccation is very real, that could be extremely problematic and extremely costly. So I'm sure there's been some studies on nest site selection and nest site preference. Again, that's not quite in my wheelhouse, but that's certainly got to play a major role. So maybe some of those quiescent periods correspond to energy reserves needed for those nesting migrations or nesting movements, or something along those lines. Certainly a hypothesis that I think could be tested, but figuring out how exactly, what measure we could use is the tricky part. But sure, there's a physiologist smarter than me that has an idea.
Yeah, something. It's interesting. I have a random question just about the methods kind of some of this stuff. So I think this is part of the ELISA test.
Yeah,
Yeah,
you're
I
good.
noticed.
Taking me back a decade ago. Just remember that.
Yeah, yeah. I don't know if this is something that you necessarily, because obviously you
Mhm.
use procedure and
Yeah.
at the time you're consulting someone, but there's a lot
Yeah.
of things that aren't necessary to
Sure.
know the details. But
Mhm.
I noticed that there's like a horseradish peroxidase and
Mhm.
like a lot of different random things being used in the
Yeah.
analysis. I'm
Yeah.
just curious, like, why is that?
Well,
Is that something? Yeah.
yeah, so it's the way that the ELISA we used for vitellogenin was an in-house designed. It was designed by my master's advisor, who's actually a lab mate of, I think, the last guest in your most recent published cast, Pamela Plotkin, was in her same lab, Dr. Roldan Valverde, who was kind of one of the famous Olive Ridley biologists as well. So he's a sea turtle guy mostly, but developed this assay to look at trachemes as like a study animal. And then we found out, hey, the trachemes vitellogenin antibody actually works for most emided turtles. So we can use it for everything. So when I got into his lab, I was like, I want to use it on terrapins. Let's do it on terrapins. He's like, well, one of my students had already just started using it on terrapins in New Jersey, so it works. So it's like, all right, let's do it. We'll do Louisiana and Florida. So it was this protocol that was developed by him and another fellow by the name of Kyle Seltzer, who basically purifies the vitellogenin molecule. And he derived antibodies for trachemes vitellogenin by injecting it into rabbits, I guess, and basically pulling antibodies that rabbits made for vitellogenin out and purifying those so they could tag vitellogenin in samples. So effectively what we do is we would have our well plates. We would plate a standard curve of samples, like known quantities of concentrations of vitellogenin. Then we'd plate our samples of plasma into those wells, let them kind of dry overnight, rinse them out, and then we put that PBS Boito, I think you asked me a question about, the PBS blotto, into those wells. Blotto, it's used like in protein assays and Western blots, right? And what blotto does is it prevents sort of like non-specific binding of other proteins. So sometimes when you put like your samples in, there's some spaces left on the wells that are open, and that can let like your testing antibodies accidentally get stuck there, and that could basically alter your concentration reading and give you inaccurate readings. So the blotto keeps those spaces closed and prevents any other remaining floating around proteins that may not have bound from sticking to those. All right, so you let that blotto sort of do its job. Then you add the primary antibody. The ELISAs we're using are called indirect ELISAs, whereas a direct ELISA is you put an antibody in that binds directly to your target molecule, whether it be, like a peptide or something like a telogenin, which is a fun word. It's a phosphoglycolipoprotein. Say that 3 times fast. Basically, it's got every major biomolecule in it. It makes up egg yolk, right? That's why eggs are so potent with energy, right? They've got all that stuff in them. But the anti-rabbit vitellogenin antibody goes in and binds to any vitellogenin that is stuck on the plate from letting it sit. Then we throw in anti-goat, anti-rabbit vitellogenin, a secondary antibody. So it's a goat antibody that was injected with rabbit antibodies. So the goats made an antibody for rabbit antibodies that can then bind to the rabbit antibodies. And that goat antibody has P&B on it. So that peroxidase. Then, or it's not P&B, HRC, horseradish, HRP, horseradish peroxidase enzyme tagged on it. All right. That horseradish peroxidase causes a color change in the samples, and that we can measure colorimetrically. But in order to get that to occur, we have to add a substrate for peroxidase. So we add the TMB solution, which is that substrate, which triggers the color change. All right. And then we add sulfuric acid to stop the color change, and it turns from like a weird blue to a yellow color. And then we run it under a color plate reader, and that basically how deep in color it is equivalent to the amount of vitellogenin that's in the sample. And that's how we actually measure the concentrations of those samples. So I hated chemistry undergrad, by the way. Absolutely hated it. So imagine my fun when I got to do biochemistry as a grad student, having to relearn all that stuff.
Well, that's an interesting question. I mean, do you think that was taking classes in chemistry, did that really help with it?
No, zero.
Yeah,
Zero.
right.
Not to discourage you to blow off chemistry, but I even took a semester of biochemistry unnecessarily because I thought it might be useful. And it was the only C I got in undergraduate, and I hated the experience that I got in it. So chemistry, I did not use any of my chemistry background. I think the only thing that was beneficial is knowing what an enzyme was, right? So, and that was stuff you learned in every other class. It's not biochemistry. Cell biology was more beneficial than any chemistry class I took for that. So
that's interesting. Yeah, that's easy.
Yeah.
Go ahead, Jack.
I said that's encouraging. Makes me feel like I'm spending my time wisely.
Well, it should help you, if you're struggling through chemistry and upset with how you're doing. Remember, I mean, a lot of people, it's not going to hit you until you're doing it and applying it to something that you're actually interested in, right? Doesn't matter how many titrations that I did in Gen Chem, like I was never going to do those that was going to be interesting, right? Didn't matter how many filtrations I did in organic chem and purifications. It wasn't going to matter to me unless I was doing it with turtles. Once you get with turtles, then you start going on your own and doing the research on it and try to understand the concept. So just get through it, man. And get through it, and you'll be better at the end.
So I'm curious about your a lot of your work was focused in like southwestern Louisiana on the terrapins, and that's something that I think we're all pretty interested in. Is like, I mean, what was it like being out in the field doing that work? And you even were finding sea turtles out there, I guess.
Yeah.
I mean, what?
Yeah.
That's, yeah.
I mean, like Louisiana, for some of its faults, is like they call it like sportsman's paradise, right? Because of a lot of the nature, and there are just some beautiful places. Have y'all been? Any of y'all been to Louisiana?
A few,
Okay.
a few times.
Yeah,
Before
Jack,
maybe.
you got to get Cody to take you up to the Comeet River drainage where he did his
No,
master's
I
work.
want to get out there so bad. There's so much I want to see.
There's so much cool. There's just really beautiful river systems. It's so great. And it's like, down here in Southwest Florida, it's like mangroves everywhere, right? It's real coastal marshes up there. So I'd never seen coastal marshes before until I moved up to Louisiana, and it's just so cool. Just I saw way more herpetofauna in Louisiana than I'd ever seen in South Florida in terms of diversity and just sheer number. Which a lot of that has to do with like the urban sprawl down here and overdevelopment of habitats. But it's really cool just to see all of the different types and numbers of those species when I was up there. Like I'd seen like 2 cottonmouths in Southwest Florida my entire life, and then moved to Louisiana, and I saw like 20 in my first week. So it's just like amazing difference to see things. 3-toed box turtles are just everywhere, absolutely everywhere. It's so freaking cool to see. But yeah, I worked with I don't know if have y'all had Dr. Will Selman on?
No, not yet.
No. He's awesome. He kind of served as like a de facto advisor, even though he wasn't at my institution at Southeastern Louisiana University, but he worked with Louisiana LDWF, Louisiana Department of Wildlife and Fisheries, and he had a long-term terrapin study going on down in one of their wildlife refuges. So I contacted him and was like, hey, I'm doing comparative research on terrapins throughout the range. Can I come out and get some samples? He's like, heck yeah, come on down. Went out on an airboat into the marsh, set some fyke nets. Also, one of my favorite parts of it was we just cruised around at low tide and went looking for what he calls mud babies. So you just find like these little potholes that were vaguely turtle shaped, and you'd poke them with a net, and there'd be a terrapin inside of them. So we were pulling those up as we were cruising around on an airboat. I was used to jumping on terrapins from mangroves down in Southwest Florida. So when I first got to Louisiana, I didn't quite understand the concept of net use, so I actually jumped off the boat onto a terrapin into the mudflats. And Will has never let me live that down for the rest of my life, but I caught the terrapin, so that's all that matters. But they're in these sort of intertidal connected marshes with variable depth that can fluctuate with tides. But it's brackish water, can get a little bit more saline at higher tides, and it can be almost fresh after major rain events because of the outflow from the main rivers. I think the Calcasieu Basin drained out into a lot of these, so in some of the upper regions it can get quite fresh. So there's a high variability of salinity, but the terrapin populations out there was just banging, just huge numbers we were catching. And they were, as Jack now has evidence of, like those megacephalic beasts too, those just monsters with these giant heads. They were so big that when I was taking blood samples, holding them up, they could hold their heads up for about 2 seconds, and then they got too tired, so they just leaned their heads down. So it was a really easy way to get blood, because I could just go right behind their heads and get it from the subcarapacial, because they couldn't move their big heads. They were so huge. But again, where they were at, sea turtles were coming up into these tidal estuaries and foraging, specifically the juveniles of the greens. I think Will had caught maybe a loggerhead as well, but we actually got a parish, which is effectively a county in Louisiana, parish record for green sea turtles in Terrebonne Parish, not Terrebonne, but wherever Grand Chenier is. I can't remember the exact parish. It was. We got a parish record. Maybe it was Vermilion Parish, of a green sea turtle we caught in a trap with about 30 terrapins. So we had a juvenile green in a trap with terrapins, and Will made fun of me a little bit because I was like, "Oh my God, it's so beautiful." He's like, "Oh, don't be a sea turtle person talking about sea turtles. We're out here doing terrapins." The little juvenile greens are really pretty, and I usually am the first to make fun of sea turtle people, but that was a pretty turtle. But it's likely that they're coming up and feeding on like the arthropods and the bivalves and things like that in those little channels, because the juvenile greens are much more omnivorous than the adults, which are seagrass obligates almost. So it's likely that at those earlier life stages they're kind of creeping up and using those arthropods to get their energy. It was really cool to see.
You sort of answered my question, but their abundance is totally fine there. Like even despite all the historical harvest, like there sounds like there's plenty of them
Yeah, they've done a lot of assessments of terrapins in Louisiana as of late. Like there was nothing on Louisiana terrapins for like 40, 50 years until Will got there and started doing his work. And Will really basically rewrote the book on diamondback terrapins in Louisiana. His initiatives with LDWF really kind of triggered that. So there is some adequate nesting habitat. I think one of Will's papers says is there's more effective habitat for diamondback terrapins in Louisiana than most other states in the southern region of the range. So there's more habitat. I can't remember the exact numbers, but there's more effective usable habitat in Louisiana. And we caught numbers. And he said the numbers we caught on my few days were low numbers. And I think we caught like 95, 100 terrapins in like 2 days. He said that's low. He's like, yeah, sometimes we're popping, a couple hundred over the couple of days. We went out in October and we're cruising around on boats and catching them. So again, we were finding they were pretty stable. There are other areas in Louisiana where their numbers are probably doing rougher, especially where there's a lot of crab trapping. Like that skull that I found, that came from a ghost trap. Like I found outside of Will's site, I went to another site a little further to the east, and I found more dead terrapins in crab traps than I did live terrapins, because there was just an abundance of ghost traps. So those ghost traps are hugely problematic for them. And Louisiana being a major seafood state plays a big role in that, unfortunately. But they are increasing regulations on things up there. I don't know if they've required bycatch reduction devices on crab traps yet, but their state herpetologist, Carrie, she's Carrie Lejeune. She's awesome, and she's doing some really cool stuff to help with the conservation of turtles there.
I guess like there's also a general trend. Like you start with the Gulf Coast, the terrapins in like the Western Gulf have like large heads that go east, and it just drops and drops. But like, where's the biggest drop off after? I heard it was somewhere around like Tampa Bay. You completely stop getting like the megacephalic ones, and they just really start getting like.
It seems like in Tampa that megacephaly is there, but then once you go south of that, it goes away. Like they're obviously still big-headed, like females versus males, right? But like and our southern populations that I work with, I think the biggest head I've found is like 35 millimeters, which is a big head for a turtle, but nothing compared to like the 55, 60 millimeter beasts I was finding up in Louisiana. So it seems like that, and I'm just starting to think about it, right? So it kind of like once you get into mangrove terrapin range, right? That's when the Rhizophararum is where the megacephaly kind of stops, and then it kind of goes back around the state. I don't know that you see a whole lot of it in the Carolinas. I haven't been up there to look for it.
Like megacephaly?
Yeah.
There's like some of it in the Carolinas, but it's limited. Like I know there's high 30s and low 40s for some of those females, but that's like really sparse. There's like totally no megacephaly in the northerns, which is so unfair because that's where I'm from. And I get everybody's like, oh, they're so beautiful. And it's like all terrapins are beautiful, but these have tiny heads and that sucks ass. So like, like, like there's got to be some sort of dietary component, but you would think there'd be a lot of overlap in diet composition in those coastal habitats. But maybe there's some specific bivalve that's in the northern Gulf that is not in the southern Gulf and up the eastern seaboard that's triggering that response. So I think we need a PhD student to do a complex dietary analysis across the range of the animals. You could even use museum specimens, right, and look at that. So it'd be really interesting. Just from like what I looked at some of the literature, that some of those northern populations were like, at least in Maryland, were feeding more on soft-bodied like crustaceans versus mollusks.
Sure.
And it's baffling to me, is even in those habitats, like mollusks, like periwinkle snails and bivalves are abundantly available, but why avoid them when that's the primary food source in like the Gulf Coast habitat?
And adding to that, like the terrapins I work with, even those are still crushing down periwinkles and crabs, but they're not showing that huge megacephaly. So what's not adding up there? Yeah, I know Will had a student that was doing dietary analysis on those Gulf Coast terrapins, but I don't think it ever came of anything. Wonder if he even still has those samples banked back. Somebody could probably go back and look at those retroactively and do some
Is he
stuff.
still actively working on dump terrapins in Louisiana?
He's back in Mississippi now. Yep, he's at Millsap College in Mississippi. He's still doing turtle stuff, a lot of stuff on the Pearl River drainage, but not terrapin stuff to my knowledge. I think him and I kind of I think we ended up writing like 3 papers from our Louisiana terrapin research, which was pretty awesome coming out of a master's, being able to hammer out 3 manuscripts from one study. So, but I think that was kind of the end of his Louisiana terrapin stuff, that work.
Talking about like gastropods, you put out a recent manuscript about the box turtle diets at some of your study locations, and
Yeah.
that was really interesting. It seems like they're eating a lot of snails too, but I'm
Yeah.
sure it's a different type of maybe more terrestrial. I mean, what was that surprising to you to find that?
I mean, finding snails wasn't that surprising. I think the amount of snails and crabs they were eating was super surprising. Like they're opportunistic, right? And these ones that we study live in a coastal environment where there are budding mangroves, right? You like I told y'all, like we can find terrapins in the same spot where we find our box turtles sometimes, which is crazy. But it's there. Why not use it? But the sheer amount of crabs and snails, I think the most predominant snail we found was called a coffee snail, which is sort of a semi-terrestrial but like sort of littoral coastal species as well. Don't know too much about the coffee snail. I think it's Melampus is the genus, something like that. But they're pretty abundant in like those sort of like coastal soils. So they're obviously foraging for them because they're available. Same thing with the crabs. At least 2 different species of crab we found in their diet: mangrove crabs and ghost crabs, or some form of ghost crab. We couldn't necessarily identify them down to species level every time based on fragments. But yeah, the amount of crabs in the diet was pretty striking, and the types of snails was pretty striking as well. Because again, I think there had only been one other paper in the literature that ever described box turtles eating crabs, and that was Gulf Coast on sort of an island. I think it was off the coast of Mississippi. There's a little herp review note. And then I mean, there have been documentations of them eating like terrestrial gastropods, like in the inland populations as well. But I mean, we were finding a large amount of those shells in their diet, and it was really interesting to observe.
Do you ever see any megacephaly in them? I'm pretty sure I know the answer.
Yes-ish. And it's where I'm actually working with Peter Lindemann on something. I don't know if he's forgotten, but I need to bother him. But he agreed to work on it with me, and now I just need to bother him about it again. Yeah, I actually went up to Santa Fe. I think I snorkeled with you up there. Was it 2 spring breaks ago with Dr. Godwin and Dr. Johnson up there. But when I was up there, I went to the UF Museum and I measured the head widths and body lengths of about like 60 of their preserved box turtles that they had. I tried to measure ones that they had like a large sample of from multiple populations, like a population, hopefully to get measurements from inland individuals to compare to my coastal and then some of the historical island populations. But there might be something going on with island or coastal box turtles where males might be exhibiting megacephalic tendencies. Not true megacephaly, like in that their heads are big, but the heads on males are definitely significantly larger than females. And again, is that related to the whole body size metric? That's where I'm going to bring Dr. Lindemann in, because he's kind of the master of those body size metrics and how that corresponds to head size. So is it just because males are bigger, thus they have bigger heads? Or is it because, hey, these males might be actually showing megacephaly for another reason? We didn't see any obvious differences in their diet in the population between the sexes down here, but it's sometimes pretty striking when you see the jowls on those big males. Just the morphology is quite different than the little-headed females.
Taggart Butterfield gave a talk in 2023 at TSA about the spotted box turtles, and those males get some like gnarly big heads, like into the 30s.
Yeah. The biggest was a 35 on a male.
That's that one he called like Godzilla or something. With this is
Yeah.
insane. But the way I looked at that, or what he was kind of thinking that was more along the lines of like combat based versus diet. But the males are like there's just intensely aggressive and competitive with each other, and I've even like just anecdotally seen that in other turtle species where it's like there's almost like some influence of just combat or age on the head size too versus diet. And those box turtles didn't look like they really had much like actual expansion or thickening of their crushing surfaces. It was just a boof-headed, like extremely large head, but not necessarily drastically different like proportions of the head. Not like a terrapin or a map turtle, gets crazy broad crushing surfaces and overdeveloped maxilla, but like just scaled up head for fighting their box.
Absolutely.
So
well, that's really interesting because we've never seen really male on male combat in our populations at all. Like never documented. Not saying it doesn't happen, but we've just never seen it. We saw one male kind of bullying a little juvenile, like subadult, but we've never seen like males going at it. We've seen male and female copulation and all that stuff, but not direct combat. We find, I think I told Jordan, we find males together more often than males and females. Like we'll find 2 or 3 males just huddled up together.
Yeah. I remember that. That's crazy.
So I don't know what's going on.
Yeah, I'd imagine like it's particularly extreme with the spotted box turtle because like at least listening to what Taggart was talking about, that he observed like tons of combat and they were fighting like musk turtles, like they were really like biting chunks off of each other, like really aggressive. And like a head width in the 30s on a box turtle is just wild, especially because like, they're not particularly big like a Gulf Coast box turtle, but yeah, that's like I'd also imagine that like he was showing like a direct correlation between I may be screwing up like I'm paraphrasing essentially between that like the head size and like aggression and the amount of territory that the males were having. And I'd imagine that makes sense. I mean, at least seeing that in other species of turtles, like okay, if you've got a male with a larger head and maybe it's more colorful, that's going to tend to be a more aggressive, more dominant turtle. So even in cap collections, like
in those, and maybe even dihydrate testosterone, because DHT can actually trigger some secondary sexual characteristics of vertebrates. So I wonder if that could be
if it's triggering head growth potentially.
It's interesting.
That's like I just very anecdotally I'd seen that's why I've been referring to it as combat head or battle head with
paper,
at least with alligator snapping turtles. Well, I call it battle head. When Michael and I went to this place in Arkansas once that was an alligator snapping turtle breeding farm, and it was essentially this pond that was the size of like a campus pond with over 100 adult macrokellies that had been crammed into it for decades. And the males were basically forced to fight each other nonstop for decades. And they were killing each other, of course, and they were all scarred up to hell. But most of those males had these crazy oversized heads, even for alligator snapping turtles. Not in the sense that they get from the diet. They were only eating fish. They were eating a soft diet. But they had the rear parts of their heads were swollen out to the side, and some of them were to the point where it was comical how oversized their heads were. But the oldest turtle had the largest head, even though the body size wasn't that crazy. And they were just all hyper aggressive to each other. Like it was basically a death pit.
Crazy.
But I was like, even from where they came from, they had these wide, round heads that the wild ones there don't have. So like that's after seeing that really got me thinking. I'm like, is the potential like is there something hormonally going on there when they're like constantly in a state of aggression? Is that literally influencing their morphology?
It's certainly possible, because those hormones can trigger gene activation, right? And that could potentially lead to some of those changes. That's really interesting to think about. So, wow, man, that sounds like a thesis or dissertation in itself, man.
Be funny dissertation at that place in Arkansas. Spend.
I think I saw was Greg Brashear that went up there with y'all?
Yeah, yeah. That place was a circus, but yeah,
That's what I've heard. He came down a couple years ago to check out some stuff, and he was telling me some stories about it. He was like, "Yeah, I had to tell some people how things are done properly in terms of handling alligator snappers respectfully." I think is what it was.
It's amazing that no one got hurt while we were out there.
It was a lot of people. Critical
Yeah.
mass. Yeah.
Yeah.
When it comes to the box turtle, you point out the differences potentially in the head dimensions and that sort of thing on the more coastal populations versus inland. How does that? I mean, like when it comes to movement and stuff. I mean, some of are they? Because we talked about this a little bit at the boxing day.
Yeah.
I mean, it seems like at some sites they'll go in the water at least temporarily and
Yeah.
cross. I mean, seawater.
Yeah. We had one that stayed in like a lagoon for like 3 weeks. Just stayed, came up for air, stayed. Why? Don't know. And then he decided to swim across it and move to another habitat. Insane. Highly aquatic. Algae frequency in this population is like equivalent to a lot of like your fully aquatic turtles. Like over 20% in some individuals. Males seem to have higher frequencies than females in terms of algae concentration, which is really interesting, which could correspond to more aquatic movement. The males tend to have those more flared shells, and some of them are even slightly depressed in their shell morphology, almost making them seemingly more adept at swimming. It's funny that when I first discovered this site, I thought, so I was looking and I saw a turtle swimming up from this lagoon, and I said, "Oh my God, it's a diamondback terrapin! I found my terrapin site. This is it." And it gets on land, and it's just a super flat male box turtle that rolled up. I was like, I just watched this thing swim like 500 yards across a lagoon where there's stingrays and bull sharks, and it just showed up. This is insane. So yeah, highly aquatic down there. That was super surprising. We had one that basically spent the winter months in a ephemeral pool for like 3 weeks from like mid-December through like mid-January. Just stayed in the water, probably because it was maintaining a stable temperature, and there were crabs and snails that could just basically pretend to be a mud turtle and eat.
You've seen some of like copulation and that kind of thing. I mean, it's some of my earliest memories from going through like Encyclopedia Turtles or whatnot are the intricate descriptions of turtle copulation.
Yeah.
And just say it varies so much. But I'm curious what that's like in the field and how. I mean, that takes a lot of observation. How have you been able to?
Yeah, it's a lot of it's just having sharp-eyed students out walking at the same time, who got used to and got really good at finding those kind of things. A lot of times I don't think we've ever been able to see the whole process start to finish. Sometimes it takes hours, right? And you're like, we're out there to do something. We try not to interrupt it. So like I've had students really say, okay, you sit and watch these two turtles for 2 hours. You're gonna sit there, let them finish their job, and then you grab them afterwards. But your job is to sit here and wait so we can process these animals. But yeah, we found them like just at the beginnings of copulation, just randomly. And while I haven't seen male combat, we did see 2 males trying to copulate with one another. So they were both younger. Don't know if they were just practicing or just got a little confused. I don't know what was going on, but yeah, we did see that once.
They're figuring
Yeah,
themselves
exactly.
out.
Just figuring things out. You got to learn somehow. And we saw him like really early in like that sort of pseudo copulation situation, and then we found him like in the typical, males lashed in, and he's just like sort of leaning back, letting her drag him around while he's breeding. We've seen that multiple times, but we haven't seen like the full thing. But yeah, just having eyes out there to enough trained eyes out there to see it is what we do. We've seen copulation occurring pretty much most months out of the year. I think there's only a few spots we didn't see it, and these were some of the times that we didn't see it. It's been documented as observed, like in Ken Dodd's Egmont Key population. So I'm sure the males are probably attempting to breed year round.
That's really interesting.
Yeah.
So the box turtles versus the terrapins, the more aquatic turtles. I mean, it seems like the box turtle season for copulation? Well, are the terrapins copulating year round? Do you think, or is there?
I wouldn't doubt it because I found males chasing females in December.
Okay.
Because their testosterone is largely a driver for mating behaviors, but a lot of times they've got stored sperm just sitting there in the epididymis, waiting to be used. So if it's there, why not use it, right? If you have the chance, like I caught males chasing females in December. One of my best terrapin catches ever was I saw a female, and then behind her were 3 males all harassing her. This was like in a typical spring, but I was able to jump down and I grabbed all 4 of them in a bear hug and caught them. It was like my best catch I've ever made in my life. It was pretty awesome. So, but yeah, they're definitely. I definitely think down here at least there's extensions. We know there's documentation of them trying to do it in like the late fall, like in the northern reaches. There was a study in South Carolina, College of Charleston, an unpublished thesis. It was Lee 2003. She documented she was actually one of the first ones to ever document hormonal cycles in terrapins. She never published her work, which is so disappointing. It did get published in this book, though. It is like the cycles that she described did get published in this book, thank God. But I used her map as sort of or her thesis as a map for my own in grad school and just expanded it to include more hormones. But she documented them in the fall, I believe, and then I think she dissected some of the some deceased males and like actually looked at enlargement of the epididymids and things like that during seasons and was able to determine like when sperm was present.
That's okay. Yeah. That's really interesting, and yeah, right. So the fact that those cycles are not necessarily linked to the act of copulation
Right.
is interesting. Like
Yeah.
you think the hormonal driver, but that's not 100%.
Right.
If you store the sperm, why not do it when you
Yeah.
have the opportunity? It's
Yeah.
opportunistic, right?
Yeah.
Well, that's interesting. Yeah, maybe something else that being out in the field a lot, you typically run across, if you've got enough experience, like you run across some interesting uh, aberrancies, deformities, that kind of thing. I'm curious what your most memorable and most interesting turtle finds are.
Can we share screens on here?
Yeah, I can do
Okay.
it if yeah.
I have a photo. So this is when I was working. I was actually before I started teaching as a professor. When I first graduated, just trying to find a job to make some money. So I actually started my teaching career as a high school science instructor, which great working with students and whatnot, but there's zero time for research, right? Except for the fact that our campus had a pond, and of course, a pond in South Florida is going to have turtles in it. And I became known as the turtle guy, right? So students, when they'd see a turtle or a snake or whatever, they would report it to me, or some of them would even bring it to me. So one of them hauled up a soft shell to my office and like, oh, it was at the bus ramp. I was like, oh, well, thanks for saving it. And then the turtle flashed me. I was like, oh, but where it flashed me was not where it should have flashed me. Like, it had another musculature opening anterior to the vent where like a deformed phallus was coming out, but it still had its normal vent. And it was just the weirdest thing. It looked like something out of like Alien. It was so odd. So it turned into a cool little note we were able to write up. So I'll send you the PowerPoint with the figures real quick, and you can take a look at it. But it's just weird. So we have a picture of the deformed one closed, where you can see there's musculature that keeps it in, which is odd, which makes me think, okay, this wasn't an injury. This is like an actual weird deformity. And then you can see like an actual what the phallus of a soft shell looks like from a preserved specimen, for comparison's sake. But it was really, really weird, but it was really cool.
Was that deformed phallus the only one present, or did it have a typical one also?
I couldn't get it to evert the typical one. This was, I think, before I'd read the paper that Ligon's lab came out with like using vibrators to stimulate turtles to evert. Also would have been really weird to have that at a high school anyway. So I didn't want to go that route. But no, it never did evert. So I don't know if that was the actual, like a secondary phallus that it developed, or if it was its actual one and it was just popping up above.
Where I'm trying to picture this in my head. Where was this opening? Was it like closer to the plastron? Was it still along the tail?
It was along the tail, but
Okay.
it was close. It was higher up on the plastron, or closer.
And like the actual cloaca was present.
The actual cloaca was present, but then there was like another opening just below the plastron on the tail.
And so, I'm seeing this now. I'll pull it up, but so there. Whoa, this is what the heck?
Can you share the photo? Michael?
Yeah, it's gonna come up. But so, okay, wait. Yeah, this is
it's weird. It's just
really
weird.
Is the actual phallus? Yeah, something. All right.
It threw me for a trip.
Yeah,
what
there
the
you
hell?
go.
So there's like the other vent, right? There's musculature there that allowed this to be pulled in and out. And then if you look at the next photo, you can see it like fully everted in comparison to a standard eversion. So it had.
Well, it looks small. Too. It doesn't
had
look
lobes, right? So what's going on there?
And this turtle was from an urban habitat. It doesn't that tracks.
Weird stuff. Yeah.
And softshells have like the scariest looking penises
of
like
any
turtle.
Can see there. It's like a face hugger from Alien.
Yeah, it's like some Lovecraftian horror. Like what the hell?
Yeah, but it had lobes, and it had like the hooks, but they weren't like fully formed. But then what made me think it wasn't an injury was the fact that the opening had musculature. Like it could choose to evert or keep it enclosed. Like it could choose to pop it out. So it was very strange, but it was really cool to see. And I think I ended up with like 3 or 4 natural history observations while working at that high school, which was pretty cool.
That's incredible. I mean, could you get hormonal profiles on that?
I wanted to, but I was working at a high school at the time. I didn't even have a -20 freezer to store things, so I couldn't.
Yeah,
unfortunately.
Dang, that is really fascinating. Yeah. It's like in a similar vein to that common snapping turtle that they found a few years ago.
You guys might be able to look at this more than me. Looking at that tail, does that look male or female to you?
I mean, it's short.
Short, right?
Yeah.
So what's going on? This is a female with 2 cloacas and a phallus? Is it a deformed clitoris? Has anybody ever looked at the clitoris in softshell turtles?
Did you do any blood work on that turtle?
I did. I was working at a high school at the time, so I didn't even have anywhere to store the blood.
Because it could be a weird intersex or
that's
something.
that was my hypothesis. Is some sort of intersex situation going on. It was very odd. Yeah.
Dude, that's insane.
Really cool, right? I'd love to catch the animal again. The cool thing is that high school is right down the road from my current institution. So if I really wanted to, I could probably get permission to trap in those ponds again.
Right. That's a project.
Yeah.
You could do a whole that's fascinating.
Yeah. We did cool enough, we did catch a Florida mud turtle at that high school too. Just one male hanging out with all of the females. And all the female striped mud turtles, that is. But no others. And I recaptured him like 3 times.
Well, why are the Florida mud turtles so hard to find? I mean, what is that? What is it about their ecology?
They're assholes. And if you're not Grover Brown, you can't find them. That's just my hypothesis. Because I don't know if you ever talked with Grover, but I invited him down for boxing turtles, a Boxing Day survey one day, a couple years ago, and he came down and then he was like, "Oh, hey, anywhere I can cruise around trying to find Florida mud turtles?" And I kind of laughed and I was like, "Yeah, you can go over here." That son of a bee drove for 30 minutes and texted me a photo of he found a female crossing the road in December. I was like, I've been looking for these here for years, Grover. Years. And you are here for 2 days and you find one. They seem to estivate for really long periods and just are really cryptic. So that is part of it. Like my friends who work with them, like a little north of me, have pretty good success trapping them. I just think I haven't found the right spot to trap them yet, even though Grover found one crossing at a spot where I had set traps. So I don't know. I just think it has to do with their inactivity, where they'll estivate for months at a time. We think they're even like laying their eggs in like their estivation burrows that they're digging, which is pretty crazy. Because even the captive folks that I've talked to with Steindachnerai say that they basically like bury themselves and lay their eggs, kind of like I think ornate box turtles do something similar, which is really interesting. So I think it's just their sort of cryptic nature and the fact that they spend like dry seasons underground and they wait for like those seasonal rains that kind of trigger emergences. Problem is when I've been really hitting my research, we've been under major drought, so there haven't been a whole lot of major rain events to bring them up. So I think I've just been missing them, but hopefully that changes in the near future.
Are they're more like ephemeral pool specialists
Yeah.
almost, like a spadefoot kind of?
Yeah.
It's
Yeah. Yeah. So like there's like almost zero information on them in the literature, right? I think like one of the most descriptive papers on them was Dodd's that just came out. It's from work that Ken Dodd did in the 80s. But he's like, somebody should probably publish something about Steindachneride so somebody can know something. So he had a couple of radio tagged ones as a mini sub project, and he followed them around for a few months and kind of measured their distance that they moved and whatnot. But they were at a sandhill pond, right? Although there were some deeper permanent water bodies in the area, he found that they regularly traveled terrestrially, and when water levels change, there's probably some correspondence to how they move, especially down here. So the ephemeral wetlands seem to be important, and those are fewer and far between down here with all the development that's happening, right? So that probably plays a role as well.
How does the development, and I mean, you've been part of some of these reintroductions of the striped mud turtles and probably some other species. I mean, what is that? How do we combat the poaching issue in Florida and
Yeah.
the development issue, and how does that impacting the turtles?
Yeah, I mean, striped mud turtles are probably not a great example for describing how development is happening, because they're like, oh, we love this. This is great. Ditches on the road. This is awesome. There's water and a bunch of garbage I can eat. This is great. They're like the new red-eared slider. So they seem to be doing pretty well with that, with urbanization at least. Like I took Anthony down to look for him, and we walked for 12 minutes and found the copulating pair of striped mud turtle so he could get his lifer when we took a walk right in a roadside ditch. Also found a stink pot, which was really cool for me. But in general, they're pretty dense in a lot of urban areas. So that's not overly impactful on striped mud turtles, at least anecdotally, right? I know Butler published a paper maybe like 5 or 6 years ago of looking at like habitat modeling and basically what climate change can do to available habitat for Cainosternids across their distribution in North America. And he said that like there was a risk of a very large loss of habitat for Cainosternid bower. Actually, I'm going to find that exact information because I was just using it to write this. I'm writing a manuscript right now, so I should have it up right now. Let's see if I find the information.
The new red-eared slider. I like that's
Yeah. But Butler's et al. 2016 is the paper, but I don't have the exact number for what the percentage of habitat loss is expected to be. But it was thought to be substantial with climate change. But they're really striped mud turtles. I don't know how much y'all know about striped muds, but they're like pretty saline tolerant. Like you can find them in the Florida Keys, and they can do fine like up to like 15 parts per thousand. So they're pretty hardy when it comes to the stuff. So that's a turtle that'll probably do well until some of the final stages of saltwater intrusion into freshwater systems, right? But poaching is another thing, right? Being available in urban areas means that any person with a net can walk along a ditch and scoop up, couple dozen, hundreds if you know the right way to do things every night, right? And sometimes, and that's where a lot of the turtles that I ended up helping to translocate and repatriate came from, were from poaching stings through joint stings with U.S. Fish and Wildlife and FWC. These were some of the few remainders that they had left over as evidence. So they wanted to try to give those a new leaf and release them and see if it was feasible. So that's how I got them. Which is a big deal. So I think all of us are turtle enthusiasts. How many of you have pet turtles? Don't be upset. I'm not judging people. I have you like Mike and
I
Jack.
have pet
You've seen
turtles.
my backyard. I have pet turtles. I've got no problem with pet turtles. Pet turtles are one of the main reasons I got into doing research on turtles, right? It's because I love turtles.
Yeah.
But one of the best things we can do is, especially like the public, is educating them what a wild caught turtle looks like versus like a captive bred. Like usually it's pretty easy to tell, especially in something like a striped mud turtle, whether it's wild caught or captive bred, right? There are some key things. Like if its shell is chewed up even a little bit, it's probably a wild caught turtle because of how connoisseurs just go ham on each other. So that kind of thing. Hey, if you're going to get a turtle, make sure you get it from a reputable captive breeding source as opposed to just finding the cheap one at the reptile show. And things like that. So I think that's a big thing, is just making sure we're educating people on the sources and making sure that the folks that are dealing the reptiles are actually being truthful about where they're getting them from, right? Because there's a lot of folks. We've all been to the Daytona Reptile Show, right? And there's some very sus individuals in terms of where they got their stock from. We've all seen those tables, right? Well, this guy has a jar full of barking tree frogs. I wonder where he got those barking tree frogs from. I'm sure he captive bred all of those. He didn't go down into one of the forests 2 days ago and just snag as many as he could, right? So I think it's just being more and making the public more aware, because folks like us, we can kind of tell, okay, that's not any good. But letting the public know where or what a captive animal might look like, and maybe being more sort of advertising heavily for like more legitimate captive breeders that we know who would be good sources for animals.
It's great that at least some of these sting operations, the animals are making it back,
Yeah.
where they belong. That's at least a good thing. But what
Yeah.
goes into that process? Because you probably have to be
Yeah.
pure genetically and
It,
in a lot of
yeah,
other ways.
it's crazy. I got to do the easy part. I just had to do the release and the monitoring afterwards. I could kind of do the fun part. But like I don't know if there was a recent issue of CCB that came out basically, and Tracy Tuberville wrote a paper basically like the anatomy of a turtle
Yeah.
confiscation. Fantastic manuscript, so good. Talks about all the challenges. I didn't have to do a whole lot of the challenging stuff. The turtles I got had been cleared to go. And they're like, hey, do you want some money to monitor these? And I said, I can't say no to turtles, so let's do it. But in general, it's a pretty complicated process, right? Because you got to confiscate. Then they've got to move through all of those transitions of source, right? Like whoever confiscates them, they're not just going to necessarily give them to the state or give them to the feds or give them to another facility, right? They've got to clear all of those chains of possession. So, and then some of them may not be releasable, right? Some of them may be just infected with gnarly pathogens, which we don't want to reintroduce into a population, right? We don't know if they had those when they were captured or if they got them from being captive with something else. They've got to clear that protocol. And they've got to be to the point where, hey, they have functional limbs. They're behaviorally able to get back in the wild, right? They're not animals that are going to be swimming up and begging for food. So we don't want to necessarily throw something like that back in the wild that may impede its success. So there's a whole lot, and there's a lot of legality to it, right? So the ones that we ended up getting down here for our release, we don't know which bus they came from. Obviously, I'm cool with that. Like, I don't want to be mixed up in any of those poaching things, just because those are some dangerous people. So we don't know which. I think they're actually a mixture of multiple bus that they had, and they were just this pool of turtles that were used for evidence that were waiting around. So they didn't tell me where they actually came from. All they knew is that they came from the region that we studied in. Didn't know exact locations, but we knew the regions. I think they got that information from some of the actual poachers or the assistants or something like that, where they were located. So FWC came to me and said, hey, we kept these 30 turtles as evidence. One of our collaborators, Kim Titterington, Swamp Girl, brought the question, hey, why don't we try releasing some of these? And then Brad O'Hanlon at FWC said, that's a great idea. Let's start the process. So they were in custody for several months, then went to Kim for another at least month, and she swabbed them and sent them out for the test for the most sort of notorious diseases like rhinovirus, turtle Fraser virus, I think mycoplasma and things like that, just to see. And they all tested negative for those things. So once they clear that step, then it's like, okay, we can probably safely release these animals without worry about them bringing more to another population. Then it's finding a good release site, right? You don't want to release them maybe in the middle of an urban park where there's roadsides everywhere, where these animals that a lot of times when you translocate animals, they go wild trying to find their old home or reestablish a new home range, and that could be problematic. It's harder to do a soft release with things like mud turtles because they are semi-aquatic, so you can't exactly build pens in the middle of a cypress swamp to kind of get them reduced to have. So it turns into a kind of hard release and frequent monitoring to see what we can do. So there's a lot that goes into it. But we were able to determine a fairly decent release site, at least we thought off the top of the head. We had I think we had like 20. I'm literally writing the manuscript on this right now, but I think we had like 20 something kind of stern and Bowery. We had like another like 10 stink pots. No, it was like 17 Bowery, like 10 stink pots. Then we had a few Steindachnerai as well, that were part of this release. So the stink pots were all too small to tag, so we just released them like in this littoral little lake edge area. It's probably the best habitat that we could have found for them in the area. And then the Steindachnerai we released like in the sort of shallow ephemeral artificial ditches, and then the Bowery we took to a cypress dome where we were actively trapping a native population of Bowery. So we introduced them where we knew we were finding decent numbers to see what we would do, and we monitored them for about 9 months because the smaller radio tags don't last as long as the big ones. And we found survivorship to be pretty high in the radio tag turtles, at least for like the 9 months. We think like 3 of them literally got murked by raccoons the week before the study finished. So if we would have finished the study a week before, we could have said 100% survivability. But of course, yeah, we found just tags laying around. So we think so they could have been dropped, but they had full scoots attached to them. So, and we know that Bowery don't usually shed full scoots, but it also could have been, hey, they just kind of tugged off as they were going into deeper areas. And we haven't recaptured any of those to confirm yet, but we did see high survivability through like 8.5 months on the Bowery. We're never able to find stink pots again, but Hurricane Ian hit like 2 months after the release and inundated the release site with like up to like 22 parts per thousand seawater. And I think that's what happened to the poor stink pots, is that they just got completely inundated by salt water. They don't do well in salt water, but the Bowery do pretty well. And the Steindachnerai seem to do pretty well with high salinity. But yeah, that led to it. So it's a big process, but some interesting, intriguing results. That at least for the striped muds, that they seem to be a pretty good candidate for just chuck and release if you find good spots.
Right. Right. Well, on the topic of, I think we'll start to kind of wind things down, but there's one more topic that we've been staring at them the whole time. It is on the topic of tracking turtles. I mean, I'm curious. So you're starting to do a lot of work in Costa Rica and partnering with some groups there, and really finding some really interesting things. Maybe you could speak to that a little and what it was like to find these guys in the
Yeah, it's the coolest. They're so cool. So I first went to Costa Rica in 2012. I was a research assistant with the Sea Turtle Conservancy. So I was cheating on what I call real turtles with sea turtles, charismatic megafauna. But in my downtime, when I wasn't looking for sea turtles, I was in the jungles looking for other herps. And on one of our first sort of morning surveys, we took the jungle trails back in Tortuguero National Park, and we found 2 juvenile Rhinoclemys annulata off the trails. And I was like, oh my God, this is so cool. So that had always been in my mind for over a decade. Like, nobody's done anything on these since the 90s. Somebody needs to work with these. Somebody needs to do it. And then 2019 rolls around, 2019 rolls around, late 2019. One of my grad school cohorts, we actually just missed each other in grad school. His name was Renato Bruno, had started an NGO in Costa Rica called Turtle Love, mostly focusing on sea turtles, but he said he wanted to expand. I said, well, what if I develop a class at my school? We bring students down to do research, and we start doing other biodiversity surveys. We do maybe some other herp surveys, turtle surveys. I have a colleague, Dr. Angus Cameron, who's a mammologist, so he wanted to do some bat surveys as well as like large mammal surveys with camera traps. So we brought a bunch of equipment down. The originally was planned to go there in summer 2021, but this was planned in 2019. The world shut down in spring of 2020, so that got delayed until finally summer 2023. We got down there for a 2-week study abroad class with 12 students, and we started just chucking traps in the water, just giving some general census of the demographics of the individual turtle species, and we caught pretty much every species on the Caribbean slope, with the exception of Conosternum and Gustapons, including some opportunistic surveys of these Rhinoclemys annulata, so that show that, hey, they're there, we can find them, let's do this. So then the idea was, okay, let's make this an annual or semi-annual thing, and in between, let's write grants to try to get down there and continue doing stuff. But the biggest part is we trained the staff at Turtle Love to do freshwater turtle work. So there is a 9 to 10 month presence of research there at any given time, where they're doing opportunistic surveys. They're looking, and when they find these Rhinoclemys, they know how to slap radio tags on them now. So they're just slapping tags on them. I think we've got 6 or 7 actively being tracked right now with 2-year tags from Hollow Hill. We got a grant from Hollow Hill for it, which was awesome. So we're going to go down and continue tracking these this summer, bring some more equipment, and kind of kick in overdrive. As I mentioned, we just got a grant from the Turtle Fest grant that Ty Park put on to hire local Costa Rican researchers to track these and collect data for the next year, which is going to be really, really, really awesome to do. So we've got the kind of the burgeoning beginnings of a new long-term project looking at the trends and demographics of the turtles, and a whole lot of other stuff that students could get into. We're going to hit diet stuff up. We're going to look at morphology. We're going to do all sorts of really cool things when we get down there this year.
It's incredible how little is known about Costa Rican freshwater turtles, for how much of a hotspot it is for research science. I mean, there's
Yeah.
like close to nothing.
The sea turtles take all of the sort of credit and information down there. It's all sea turtles, right? But the freshwater turtles, like the moles, were the last ones to really do work there, right? And I think there was maybe a little bit of stuff out of La Selva on some Rhinoclemys spunaria stuff done like in the mid 2000s, but really just like really minor, not minor, but smaller sort of contributions of like demographics and things like that, but not just nothing. So it's literally a blank slate, and that's kind of how Southwest Florida was for me when I started too. It was kind of a blank slate when it comes to like reproductive stuff. Costa Rica is the next blank slate, and we're starting to write the story. And the biggest, most important part of that is not just doing, like parachute ecology, right? Jumping in, going down, throwing traps in, then bailing, right? Training and working and collaborating with local Costa Rican folks and institutions to continue the project, to eventually, the hope is, they'll eventually kind of take over running these things fully. We'll still come down and visit and contribute and bring students down for the experience. But the hopeful idea is to get these conservation projects running full time in country, as opposed to us coming down sporadically to do a lot of it.
That's really exciting. Yeah, right. Get the local people involved, and that's how you're going to get great data and foster the community around it for sure.
Exactly.
With the Angustapons, the search continues for them, I guess. I mean, what have you learned just through talking with people there?
Yeah, I have some information, but I'm not going to share it yet, just because it's super secret. We are hot on the trail though. But in general, they seem to be like Steindachnerai, where they're number one, the bane of my existence because I can't find them, and number two, they seem to be highly seasonal. And like I think you and I have talked about like the Legler paper from like the 60s, right? Like with Legler 61, I think it is like the only like really descriptive paper on Angustapons. And it's like, hey, we caught like 8, and that's all we know. They lived in this like little oxbow. But we've got some local folks that have claimed to have seen them, and they may have some better ideas on the habitat description that they may be using. So we're hopeful we'll be able to do that. We're actually kind of partnering a little bit with some folks with NAFTR that are going to come down and do some trapping this March at some spots to see if there's anything going on there. But then we'll hit it really hard this summer and see what we can find.
That's awesome. That's exciting. Yeah, that's one of the most enigmatic turtles. It just doesn't. But again, like that could just be that we haven't keyed in on their ecology whatsoever. So
Yeah, that's why if we can get some radio tags on them, that's gonna change the game. We'll be able to blow it wide open if we can just get a couple of tags on them.
right, right. Well, as we start to kind of wind down, I'm just curious. I mean, a key theme throughout your pursuits is just this creativity in it. Like presented the opportunity to go down to Costa Rica, like I'll just make a class. That's a really unique, creative thing to do. How do you view projects? I mean, are there any? Is it just the passion for it, or
Yeah.
yeah,
Yeah, largely, I just love turtles, and any chance I could work with turtles, like I will find a way to work with turtles, right? My initial driver into turtles was like when I was an undergrad, my like I'd always wanted to work with reptiles, right? I wanted to be a herpetologist since I was tiny. After paleontology wouldn't pan out, right? But when my undergrad professor Phil Allman mentioned that reproductive cycles could be different in southern turtles, I said, all right, that's what I'm gonna study for the rest of my life. I'm right there in that class, and that's what's sent me on this trip. And then as I've kind of gone in and expanded, because reproductive physiology isn't necessarily applicable to a whole lot of things, right? It's really expensive to do it. So then it comes into, okay, what can I do with a limited budget? There's always market capture, demographic studies, but diet studies, they're easy to get samples for. Students can be trained in it very simply, right? And they can conduct research in a short period of time. So a lot of it has to do with what kind of projects can I at least start in a year period when I have my research students with me. And that is one of the main drivers. Then it goes on to there, okay, as we're going out and documenting things, natural history is huge, right? When we notice little noteworthy things, that is something that could expand into a bigger study. So we kind of build upon the smaller observations we make and kind of go with it as it comes to us. And the ideas just kind of form themselves. We move on from there.
that's all. And the emphasis on natural history is great. Nowadays there's so much. I think sometimes things get lost in the computational aspect and
Yeah.
a lot of
Everybody likes the omics and the analytics, but
yeah,
there's so much in the natural history that needs to be understood. And a lot of that will play directly into some of the more complicated analyses that we could do on these animals. So we can't forget natural history. It's still extremely important.
right. And so one way that I mean, I'm curious of just the most inspiring moments in the classroom and teaching and that sort of thing, and you're bringing a lot of this information together too at
Yeah.
the Herpaton meeting and that sort of thing. I mean, what have been the highlights from just seeing the students engage with this sort of thing?
Yeah, I'm like a proud papa when it comes to my students, like especially when they present posters or talks, like I have to like fight back tears every time because it's such a proud moment for me, understanding that, hey, I've gone from being that student to now to the position where I'm able to help students expand their horizons in that matter. It's so cool. But some of the coolest has just been the fact that I've been able to facilitate like undergrads, sometimes like 2-year undergrads, right? Because my school is largely an associates driven institution, similar to Santa Fe. Like I've got students as lead authors on publications coming out of a 2-year institution before they're even hitting their 4-year degree, and just seeing how that allows them to build and expand. And some of the places my students have been able to go after my class is it tickles my heart. Like I'm just like, this is just so cool, and I'm so happy to go play a small role in you being able to achieve that.
That's awesome. That yeah, and it's a cool way as a professor to make an impact that expands way beyond. You can do the thing you love and you just recruit people into it and
Yeah,
get each other done, and then
yeah,
it's beneficial for everyone, right? That's huge.
yeah. Grover Brown says I'm spreading the good word of the cult of the turtle. So keep indoctrinating the turtle cult.
That's a good way to do it.
No,
Yeah.
you and Grover are very good at
Yes.
that. That's for sure. I've been like I mean, social media was kind of something with that. I've been like asking like turtle questions to you and Grover for years, like since I was in like high school and like younger
And
than
he's
that.
so is he
Like,
not like the nicest guy with responses to people? Like he will share.
oh yeah, it doesn't matter who you. He's like, if he sees someone that's got an interest or passion for turtles, he's like, all right, I'm gonna recruit. I'm gonna recruit them.
100%.
I'm gonna brainwash them with
Exactly.
turtles, and that's what they're gonna do.
That's I think we've all got a similar story with Grover and yeah, such a nice guy. I mean, so we're always curious just in terms of all your field work. You know, everyone's got crazy stories. I'm curious if there's anything that stands out in particular.
Oh, I mean, a lot. I'd never had a broken bone in my life until I was doing my terrapin work. And I was doing mangrove parkour is what I called it. That's my capture technique, or real turtle rodeo as I call it. You know, like the sea turtle rodeo where you jump off a boat onto a giant turtle. That's easy. Jumping into a log-laden mangrove creek, a little more difficult. So a male terrapin was coming through this channel, and I was perched on top of a mangrove like a freaking gargoyle. I was just waiting for it, and I jumped on it. I did not see the giant submerged, broken black mangrove log as I dove in, and I went in and I shattered my hand on it as I punched. I got what's called a boxer's fracture, which is apparently super common, not from boxing. It was first described in boxers, but apparently it's the most common injury for meatheads at the gym punching the wall after missing a layup. Which is what my doctor accused me of doing because he did not believe my turtle story. So that was one of the worst that I was in a cast for like 6 weeks post smashing it trying to catch a terrapin. So that was one of the craziest. But one of the cool, crazy, scary ones was actually in 2023 in Costa Rica. We were tracking some new trails, looking for some spots to set traps and whatnot. We kind of got a little disoriented into where we were, and the sun was starting to set. And we're like, oh, this is getting a little bit touchy. Okay, but we could hear the ocean. We knew it was close. We just didn't know the best way to go. And then at the same time, an entire colony of army ants started moving across the forest floor and cover the forest floor. So we had to literally just sprint through this dense forest floor covered in army ants, not standing still, because if you stand still, army ants will crawl up you and it would not be a good time. So we had to literally just haul ass, not even thinking about the fertilances that are likely sitting around, or the palm vipers that may be hanging out in the area, and get out of there in limited sunlight with these ants. And we all made it out fine. But yeah, there was I think the saying of our trip became don't stop moving, run. That was that was it. So that was pretty crazy.
There's something different about Central America. I mean, some of the things, like, just any number of issues. Like in Belize, like I can relate to that. You know, just any kind of. We were trying to hack through to make a trail somewhere, and a Tayra jumped out. And it wasn't dangerous, I don't think, but it was going right towards me, and I jumped out of the way, and and it's like everything out there is potentially dangerous. You know.
Yeah, we had a herd of like 70 peccaries come strolling through, and it's like, oh Jesus, they have babies with them. They could get really pissed. We could get stampeded on, so we had to be ready to jump up a tree if we need to. It's different down there. It's awesome, but it's different.
The peccaries are the things you got to look out for. I mean, according to everyone, that's the stuff that's, if they're charging, I mean,
they'll mess you up.
Yeah, that's
much more worrisome than jaguars for me.
Right. They don't want to be seen, right? No. Yeah. Well, I guess just to wrap it up, I mean, we're always curious as students, and what advice would you give to someone that's looking to make turtle work a career?
Yeah. Number one is, don't be afraid to stray away from your objective in order to get back to it. It's really hard to find positions in academia or in the job market that necessarily directly relate to turtles. Don't be afraid to stray off that path a little bit and study stuff maybe adjacent, aquatic ecology, get an aquatic fisheries job. that could lead you back to turtles. I think that's a big thing. I was very lucky that I never had to do that, so I can't say squat. I was just lucky, and I was just stubborn. I just kept sticking with turtles. But a lot of times it doesn't work out that way. So don't be afraid to kind of stray elsewhere and stay adjacent to your turtles, but don't be afraid to expand into other things. And then the most important thing is to volunteer and make connections. Go to those conferences. You'll meet nice people. Go to the Herpeton Florida Herpetology Conference this year on November 1st and 2nd in Naples, Florida. It's officially happening in Naples this year. Sorry, I had to throw a segue there. Yeah, go to those conferences. Meet people, meet potential advisors, funders, etc. That's the best thing you can do, is to make those connections. Those people will remember you, and that can lead to more opportunities. Michael, you've been presenting at conferences since you were like 14, right?
Yeah, it's been a while.
Yeah.
I'm going to one this weekend. It's like the MIT AJS AAAS thing,
Awesome.
and I had to. It's talking to a bunch of students in the
Oh, cool.
academy, and I was tallying that up, and yeah, it's been a while. Like I've been incredibly fortunate with all those opportunities.
Awesome.
And just
Pay it forward, man. That's beautiful. Love that.
Yeah, it's exciting for sure. It's going to be a big crowd though, so it's going to be interesting. But yeah, but I mean, we'll take that advice to heart. That's really not everyone says that, but I feel like a lot of people maybe have done that and they don't realize it, and so it's good to hear that. Well, that's awesome. I mean, we like to finish off with a little turtle trivia. I think that you've got a pretty good chance of stumping us. So I don't know if you've got a question, or should we hit you with one? However you want to do.
I'll hit you first. I'll hit you up with kind of easy one.
Okay.
I'd say kind of easy, but it requires a little bit of like historical knowledge. What was the first species epithet for diamondback terrapins, and when, and who?
Yeah, that's
a tough one.
Yeah, I don't know. I don't think I have that one.
Know anything about the University of Maryland? What's their
Little bit.
mascot named?
It's a terrapin.
He's got a name.
Like, what is? I don't remember the name. I should.
What is like the default original genus for turtles for the most part? It's kind of gone away. It only represents old world testudo. Yeah, testudo terrapin was the original epithet. 1793, I think. Shope. Yeah, was
Shope. Okay.
Yeah, I was trying to think when I had that date in my mind.
And then I think it was like early 1800s when Gray moved into Malaclemes. So, but
that's a good one. That's simple and eloquent. You know, like that's
yeah. I didn't want to hammer you with some reproductive cycle stuff. I think that would just be not fair, because it's such a niche, even within a niche field, to know reproductive cycles and hormone triggers.
Yeah. I mean, if you've got something that's crazy, we could try.
Okay.
But, if not, no.
I mean, considering how we fared with that one, I think
that's fair.
When focusing on the morphology of the carapace of Aldabra or Galapagos tortoises, focusing on peripherals, a key difference between the two is the presence of what?
Of the peripheral scoots you're talking about, like the bones?
Yeah, like I mean, scoots or bones work. Same difference for this.
You know, I have always had a problem just looking at Aldabras and Galapagos and telling the difference between the two, because they just both look like big drab tortoises to me. I love them, but they just look like big drab tortoises. Shoot, I would have no clue, because osteology is not my my forte.
No, just the I was talking about the presence of a nuchal scoot. The
okay. So you're just talking. So is that considered a peripheral then?
I would consider it a peripheral, right? Because it's in that row of. I don't know.
It's second, but I was gonna say something to do with the nuchal scoot, but but I wasn't sure if that was
describe that better. But
no. Right. How do you tell a gopher tortoise from a sulcata? That's a common question we get down here in South Florida. Because we get so many gosh damn loose sulcatas roaming around, and usually small ones.
I mean, if one's like 100 pounds, you can
Yeah.
usually.
Well,
Yeah.
it depends
Yeah.
on what Jimmy down
Tie.
the street says, because he tells me that it's just the biggest gopher tortoise on record,
Could
even
be.
though it's clearly not. But any ideas on a quick reference for whether it's a gopher or a sulcata? If you
Maybe
were
the
to
spurs on the front legs.
Sulcatas have no
boom,
nuchal
nuchal
suit.
scoot again. Yeah, no nuchal on sulcata.
Dang! Look what happened with that one. Took the same question.
Put it around.
around.
Yeah.
The
Yeah.
same
Although those
question.
ones, like, I mean, what's really the relationship between a sulcata and a gopher tortoise, right? There's like zero relationship other than that they're tortoises.
Also, I feel like using that as a diagnostic for
Yeah.
general people that aren't
Yeah.
familiar with turtles would
Yeah.
not be super
No.
helpful. Right?
What? You losing the what?
Yeah,
Yeah.
yeah, right.
Yeah. But no, I commonly get people messaging me, I found a sulcata. And it's like a baby gopher tortoise. And I'm like, no,
No.
no, please put that back.
The duality of
Yeah.
man. That's the best.
We need to find a home
They're
for
like,
it.
hey, this is the biggest gopher tortoise
Yeah.
I've ever seen. It's just a big
Yep.
sulcata.
Like if you go on to like some of those tortoise Facebook groups or like wildlife, it's so
bad.
insane. It's like, I've had this tortoise for 30 years. It's my sulcata. No, it's a gopher tortoise. Why is he so small?
What's
Yeah.
what before we hop off? What's the craziest non-native turtle situation you've seen in Florida?
Cool,
That's
some cool stuff, man. I mean, basically 90% of the turtles I have in my backyard are ones that have been found roaming around Southwest Florida. So, I mean, I was doing some herp surveys at a park and I saw a weird tortoise walking from about 200 yards away, and I was like, that thing's walking like it's got somewhere to be. That's not a gopher tortoise. That's a Russian tortoise. And I sprinted, and by God, it was a Russian tortoise walking to a gopher tortoise burrow. Just there. Just I was like, I knew it was a Russian tortoise because it looked like it was walking with purpose. So that was pretty crazy. We've got some instances of what is it? Paleomedusa down here just crawling around. A guy called me. He's like, I found a bunch of stray cats beating up on this turtle in the middle of the street. What is it? It's a damn Paleomedusa, I think. Yeah, I can't remember. They've changed the taxon. I think they're ventralis now or something like that. Paleomedusa ventralis, the West African side neck terrapin. So yeah, so those. We caught a Graptomys watchatensis in our traps at the school where we host the conference in our traps. A male that was like the biggest male Graptomys I've ever seen. It was like the size. It was like, I think it was like 11 centimeters. It was huge for a male. I had no idea what was going on. And yeah, so that was a big one. I feel like there was at least one other major one. I mean, the redfoots I have, most of those were found just crawling around. The leopard tortoise I have was found crawling around. So yeah, it's just a matter of time before something else gets established down here. It probably has already happened and we just don't know yet.
You've already got red ears and Mississippi maps established in breeding in some areas. So.
Yep.
Yeah.
Yeah, I think it's there's a Claudius species on a zoo property in Miami, I think, that's reproducing.
It was Starotypus.
It was Starotypus. That's what it was.
Yeah, Cypricatus.
That's what it was.
Salvini too. It's I think Salvini there.
Yeah. So yeah. And there's been reports of Rhinoclimes funaria in that same area, so I'm sure they're around having eggs. So it's just a matter of time.
I was just recently updating a bunch of turtle diversity metrics for these southeastern states, and it would be interesting. We didn't factor in non-natives, but I actually think you could, in Florida, make a case for it being the second most diverse country, just the state of Florida.
If you take in, I mean, our invasive lizards outnumber the natives by like exponential factors. That's the sheer amount. Turtles will take off that way too if it happens. I wrote a little descriptive note on Russian tortoises in Southwest Florida because I had like a documentation of like 9 or 10 reports of them. Most of them are in my backyard now from people that have found and collected them. Just it was really crazy. We had like a random, like, well, not random, but like a really high drought season. And I went out to my enclosure and I found a hatched egg and hatchling, naturally ground hatched Russian tortoise. So it's like, oh, it's possible if the conditions are right. I mean, if you'd require like really dry wet season, like we had like less than like an inch of rain from like May to July, when this animal presumably laid its egg, and it just hatched naturally in the outdoor enclosure. So I wrote a little descriptor on that with general reports of the Russian tortoises that I'd found and vouchered. So yeah, it's tropics, stuff can happen. My guess, my hypothesis is the next big introduced one that will boom will either be Geoclimes, the Hamiltoni, or maybe one of the Palomedusas or Pelusius. One of those I think will become established just because of how many people have an overabundance of Geoclimes in South Florida, because you can't sell them across state lines, right? Because of the CITES status, right? But then like the Pelosi, just because there's so many at pet stores, I feel like they're going to be like the next dump turtle, but we'll see.
Right.
Yeah, that's funny you say that. I'm in New York, and I've still found side neck turtles dumped, and like that's just like seasonal because obviously they're not going to overwinter, but like it still happens here.
Yeah, it's crazy.
Dang. Well, all right. Well, we'll sort of finish up here. I think if there's anything that you'd want to leave off.
I guess one more thing
Oh yeah.
I'm going to say. On the topic of dumped turtles, this popped back in my head. I found a western painted turtle in Delaware one time, which is obviously not where they're supposed to be. And I had like just glanced at it briefly. It was at a public park, and I was like, "Oh, I think this is just really random. I'm just not remembering it." But I was like, "This doesn't seem quite right." I just assumed it was a juvenile red-bellied cooter for a second, not really looked at it. And then I think Michael ended up with photos of it or something, and he's like, "That's like an old male Western painted turtle." And I'm like, "Wait a second, what?" And I went back and caught the turtle again and actually like looked at it, and I'm like, "I'm an idiot for just assuming this is a juvenile without looking at it." It's like kind of embarrassing because it's like an old, worn down, like clearly wild, like it almost looked like somebody went out to like way out west, caught a male Western painted and dumped it in the. It had like the most classic like Western painted plastron and coloration. It was just, that was one of the weird ones because I'm like, that's not one that's like often in the pet trade, I feel like. And why would somebody dump that one so far outside of its range? And like, that was weird.
That is weird.
That was
really
just
weird.
That was one of the strange ones. Like sometimes you never know, like you'll find something that's just, it's not a slider or a Mississippi map or a redfoot. It's like, what
we found an eastern painted turtle dumped in that Gopher Tortoise preserve that y'all visited. Yeah, where there's no standing water.
Most people shouldn't have turtles, man. Like it's so bad. Like that's like the thing with sulcatas and red ears, especially. It's like those should have never been as easily available as they are.
Sulcatas still available as easily as babies.
It's a disaster. Sulcatas are like livestock. Only people that like you should have to get some sort of permit or something, or take a class before you get one of those. Like that shouldn't be like, oh, I can get one for $50. I can get a hatchling tortoise that can grow to the size of almost a Galapagos tortoise.
The third largest terrestrial species on the planet. It's like, and you can buy them for $50 at a flea market. Come on.
You get the right lineage. You could the right lineage, and it's got enough food. You're talking about a tortoise that could be 300 pounds and is digs giant burrows and bulldozes.
Tell me about it. You saw the burrow of mine in my backyard.
Yeah.
I think we got in the burrow. Yeah.
Yeah, I tell you, I agree, concur completely.
Yeah, well, I think that's a good place to kind of finish up at. But if there's anything you want to finish up with, or
no, just thanks again for letting me on. I don't know when y'all will release this officially, but always wanna hype the conference up. Yeah, we're having it that basically Halloween weekend. So the weekend after Halloween is when the conference is going to be this year, and we're going to be hosting it at the Naples Botanical Gardens. This is the first official release of that. So whenever this one comes out, hopefully we can spread the word about the Herp Conference. So if you know anybody that wants to attend a really cool sort of low-key Herp Conference that's still got some high-quality information, come on down to South Florida, and we'll treat you well. Just, if you're a turtle person, you might fall asleep in between sessions, like somebody I know who might be on this
classic.
But anyways, yeah, definitely come down and check it out. And if you have ideas for sessions, feel free to submit them to me.
Awesome. We'll put a link to it in the description. It might be a little before it's out, but we'll put it out on the social media and everything.
Awesome.
Sure. But yeah, we all hope to be down there. Awesome. Well, this has been great. Thanks again, Jordan. This has been awesome.
Appreciate y'all having me on. I really
awesome.
appreciate it. It was a lot of fun.
All right. We will see everyone in the next one.